Okay, welcome to the last lesson of DNA replication. And so the last part of this lesson is going to talk about why chromosomes get shorter every time DNA is replicated. So it's going to start with me explaining it, and then there's a video that I found on YouTube that explains it in a little bit more detail in case you're interested. And it also explains the leading and lagging strand thing again, so it might be interesting for you to watch it again. So once again, here we have our model of DNA replication. We see this side, this here we have going in this direction, and this going in that direction. Notice that this one is opening towards the fork, so this is the leading strand. Being synthesized towards the opening fork, so this is the leading strand, and this one's going in the opposite direction, so it's the lagging strand. Okay? Now, at the end of each of these, okay, and on this side you don't see on the lagging strand you don't see the Okazaki fragments yet because this hasn't opened up. But you'll see on both strands, there's this red part, and that red part is the RNA primer. Okay? And if you remember, what happens is that DNA polymerase I removes this primer. Now, when this is removed, okay, so I'm actually going to delete it. But you remove this, that's one RNA primer. As it doesn't delete. It still doesn't delete. There we go. And when you move this, we have that problem again, that RNA polymerase, or RNA polymerase 3, so it's removed by RNA polymerase 1, but we have this problem that RNA polymerase 3 cannot add to nothing. Like, there's nothing to add here to here. So, okay, cells have evolved a solution to this. Otherwise, what would happen is that the chromosomes would endlessly shorten, and death would occur pretty quickly in cells. So the point is that at the 5' ends of DNA, the DNA gets shorter after every replication. Okay, now, how have cells solved this problem? Okay, the ends of chromosomes, okay, as we let this load, whoa, the ends of chromosomes have non-coding sequences that don't code for protein, and we're going to learn that DNA codes for protein. So basically they have drunk DNA at the ends of the chromosomes, here, okay, that if it gets cut off, it doesn't matter, it doesn't do anything. It can last about 50 cell divisions, until it's completely degraded. But it's DNA that is just protecting the chromosome. It doesn't code for anything. Additionally, some cells have an enzyme that's upregulated or that's active called telomerase. And it's the job of the telomerase enzyme to make the telomeres longer. Because it has the ability to add bases to when nothing is there. Like, it doesn't have, that DNA polymerase 3 does not have that ability, but telomerase enzyme does. It can make those telomeres longer. Now, just relating back to something that we learned in the previous unit, in cancer cells, one of the things that is turned on at high activity is telomerase. It's activated at a high level. And that seems to be a marker of cancer cells, that telomerase is highly active. And the reason why that is, if you remember, in cancer cells, they're dividing quickly. So they need their telomeres not to shorten, otherwise their chromosomes would be gone and the cell would die. So in cancers, telomerase, the enzyme that extends the telomeres, is upregulated. It is also high in stem cells, but we haven't talked about stem cells yet, so we'll leave it at that. So what's going to happen now is I'm going to play this video which explains this in a little bit more detail and also talks about the leading and lagging strand again. And then on the homework about DNA replication, there are some questions about telomerase on the homework. So I will see you guys after Thanksgiving break.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction, and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.